This is DNA. DNA is the genetic material of a cell that is used for protein synthesis. It is found in the nucleus of a eukaryotic cell, as will be shown in this video, or in the cytoplasm of a prokaryotic cell. This is a gene. A gene is a section of the DNA that contains the code for the RNA during transcription that will eventually lead to protein synthesis. So let's talk about the first stage of protein synthesis, transcription. DNA is coiled in a double helix shape. It is not coiled here for an easier visual. DNA is made up of the sugar deoxyribose, a phosphate backbone, and nitrogenous bases. The bases are thymine, adenine, guanine, and cytosine. Adenine pairs up with thymine, and guanine pairs up with cytosine. There are 3 stages to DNA transcription: initiation, elongation, and termination. Initiation starts when RNA polymerase binds to the promoter. In bacteria, there is a promoter on the DNA. In eukaryotes, there is a ta-ta box, shown by the nucleotides. Several initiation factors are needed in eukaryotes for initiation to begin with RNA polymerase. The next step is elongation. Elongation is where RNA polymerase goes and makes an antiparallel RNA strand to the template strand. The template strand is the one that is used in RNA polymerase, and this is the coding strand, which is not used. RNA polymerase goes down the template strand and makes an mRNA strand that is base pairs of the nitrogenous bases of the template strand. Uracil is used instead of thymine because there's no thymine in RNA. The final step is termination. This occurs when a DNA sequence in the template strand causes the RNA polymerase to stop making mRNA. This pre-mRNA detaches with the RNA polymerase, but it stays in the nucleus for modification. To see modification, we need to zoom out. This is the same pre-mRNA for RNA modification. Pre-mRNA is made up of 2 main parts: the exons, or what is expressed, and the introns, or what is not expressed. There are 2 untranslated regions at the end of the pre-mRNA. A 5' cap is added on to one side, and the 3' poly A tail is added on to the other side. RNA can work with proteins like enzymes to cut out the introns from the genes. This is called a spliceosome. The exons are then spliced together. RNA splicing can occur in many ways to make different proteins. Now there's the 5' cap, the untranslated regions, and the exons, and the poly A tail. This is modified mRNA, or messenger RNA. The mRNA then exits the nucleus through pores in the nuclear membrane. The 5' cap and the poly A tail help protect the genetic material from hydrolytic enzymes and also help attach to the ribosome in translation. Now the mRNA is in the cytoplasm where translation begins. Protein synthesis is when proteins needed by the cell are made from the genetic information stored in its DNA that is made into RNA in transcription. Translation begins when the large ribosomal subunit, made up of rRNA, or ribosomal RNA, and the small ribosomal subunit attached to the mRNA in the cytoplasm. There are 3 steps to translation: initiation, elongation, and termination. Initiation starts when the start codon, AUG, is found. We have to back up for a minute to explore tRNA, or transfer RNA, that continues the rest of protein synthesis. This is tRNA, or transfer RNA. It is made in the nucleus. There are many different types of tRNA, each with a different anticodon. This one's anticodon pairs up with the start codon, AUG. tRNA has a 3D shape thanks to the hydrogen bonds between its base pairs. tRNA then goes to the aminoacyl tRNA synthetase to get its amino acid that corresponds to the codon. In this case, that's met. The tRNA forms a covalent bond with the amino acid. Then it comes and enters the A site on the ribosome. There are 3 sites on the ribosome. The A site, or the activation site, is where the tRNA enters and pairs up with the codon. The P site, or peptide site, is where the polypeptide chain forms. The E site, or exit site, is where the tRNA leaves the ribosome. The tRNA enters the A site, and its nitrogenous bases pair with the AUG start codon. This codes for methionine, the first amino acid in the polypeptide chain. A codon is a group of 3 bases that together encode for a specific amino acid. Each codon has an anticodon that it pairs with for the tRNA. The start codon, AUG, sets the reading frame for the rest of the mRNA. This means that these 3 bases will be a codon together, and it can't be these 3. The tRNA enters the P site, or peptide site, and is ready to start building the polypeptide chain. The second stage, elongation, starts. Next, tRNA enters the A side. The amino acid that is on the tRNA in the P side forms a bond with the one in the A side thanks to peptidyltransferase. These 2 connect. Peptidyltransferase is the enzyme that helps form the polypeptide chain. Finally, the tRNA that is in the P site but no longer has amino acids attached moves to the E site, and the tRNA that was in the A site that now has the polypeptide chain moves to the P site. The tRNA that is in the exit site then leaves the ribosome and goes back to the amino acid tRNA synthetase, where it begins again. This process continues until it reaches a stop codon in the mRNA. Next is the final step, termination. At the end of elongation, there's a stop codon in the mRNA. This stop codon sends a signal into the A site to stop elongation. This signal hydrolyzes the bond between the tRNA and the polypeptide, and the polypeptide is free to go into the cytoplasm. The final tRNA then enters the E site and exits the ribosome. Amino acids are the smallest units of a protein. Together, they make up the polypeptide. The polypeptide, if it is in a bound ribosome, leaves the ER and goes to the Golgi, where it is packaged and sent out of the cell. And there you have it, protein synthesis from the start to the end.